Great. Can everybody hear me? Yeah, I think so. Welcome everybody. Thanks so much for inviting me. Thanks so much to the organizers. I'm very happy to be back again. I think the meeting in the next 2 days was scheduled for 2020, so it's a long time ago. We met here last time in 2019 at the same meeting. I would like to share some of our recent work with you, and I'm a cognitive neuroscientist, so I'm interested how higher level cognition is implemented in the human brain. We have a very strong focus on memory. I find memory fascinating. Remembering all the events around us, they are so important for us. They shape us. They shape almost our personalities. Remembering all the details of our daily lives. And when we talk about memory, of course, as a neuroscientist, we need to talk about the hippocampus, the hippocampal formation, the hippocampal system. And when we talk about the hippocampus, we should talk about spatially tuned cell types in the hippocampal formation going back to the 60s of the last centuries with the advent of electrophysiological recordings in the freely moving animal, pioneered by many people. I only mention here John O'Keefe and the Mosers, and it's a huge field developing over the last 50 years. So what those researchers discovered are spatially tuned cell types in the hippocampal formation. When you record from single cells or single units, or now populations of cells with new techniques from the hippocampal system, you see a map-like representation emerge. So you see the famous place cells in the hippocampus. So those are cells which signal a location of the animal. If the animal would forage this room here, one place cell would fire over here, another place cell would fire over here. And in sum, what we assume is that the whole population activity of place cells would give rise to a map-like representation of this very specific environment. Some people would call that coding regime the most fascinating coding regime outside the sensory cortices. When you bring the animal back into the same room a few days later, the cells would recognize the place. So the same place cells would fire at the same positions, giving stability, which is, of course, an important feature for a memory system. When, however, the animal enters a new lecture hall, a new room, different cells would fire at different positions. So the cell population can distinguish between different spatial contexts. You also have then discovered by the Mosers in 2005 the famous grid cells, which are only one synapse away from the hippocampus. So the grid cells are located in entorhinal cortex, more precisely in medial entorhinal cortex, and they are largely in the upper layers, layer 2, layer 3. In contrast to the place cells, where a single cell, this is a schematic of a place cell, only indicates a single location in the environment, the grid cells, a single grid cell would fire at multiple locations, and in this very specific characteristic hexagonal firing pattern. And of course, these cells are not alone in the system. There are other cell types. It's a whole slew of spatially tuned cell types. You, for instance, have border cells or boundary vector cells which encode the presence of environmental boundaries in the environment. You have object vector cells which encode the vector to an object in the environment. You have speed cells encoding for the speed of the animal and so forth. So together, that's why I call it a space machine, these different cell types, these different coding mechanisms provide a map-like representation of the environment. I'm a cognitive neuroscientist, what I said at the beginning, so I'm interested how this map-like representation is working in the human brain, supporting higher level cognition. And we formulated a couple of years ago, building on work of many others, some ideas of how the system, the hippocampal entorhinal system, based on these spatial coding principles, would give rise to a machinery which could span other spaces as well, which could be relevant for higher level cognition. Let me just illustrate that at this simple two-dimensional feature space, a vehicle or car space, where on one dimension you have the engine power, on the other dimension you have the weight of the car, and what you see is that, for instance, a lorry is located over here, so high engine power, high weight. A Formula One racing car is much lighter but has an equal or even stronger engine power, and so forth. So, and these cognitive spaces help us to organize experience, representing information, high-dimensional representation in low-dimensional representational spaces, if you like, and can support encoding of information, structuring information, conceptual knowledge acquisition, and of course also decision making, because you need to somehow draw from these representations when you make decisions. Of course, that builds on previous work going back to Thoman in the 40s of the last century with his development of the cognitive map theory, and the idea then revisited in the context of the discovery of the place cells by John O'Keefe and Linda Dell in their famous book 1978, and then of course bringing these ideas outside the spatial domain by Howard Eichenbaum and others. So in sum, what we think, and many in the field think is going on, is that the hippocampal entorhinal system provides a low-dimensional representation of cognitive variables. And you can also, of course, quantify that with modern techniques and understand these neural state spaces, these low-dimensional representations of environmental features of cognitive variables. There's a lot of experimental evidence, in particular when you look at these neural space representations in various domains, not only spatial navigation, but also memory decision-making, where people also want to understand the neural geometry. I have that as a discussion point for the general discussion, understand these neural manifolds. And there's also, I would almost say, a boom in computational ideas, computational models trying to describe somehow the development of these state space representations. Just to name a few, I will in brief mention the Thoman-Eichenbaum machine by Whittington and Tim Behrens, but also their ideas about successor representations and many other ideas. So let's maybe briefly summarize the Thoman-Eichenbaum machine from Tim Behrens' lab, I think, because it nicely summarizes the computational principles in the hippocampal entorhinal system. So they try to integrate these different ideas within a computational model and unifying these hippocampal roles for spatial and non-spatial inferential processing, if you like. And the model suggests that the hippocampal entorhinal representations enable generalization by their factorized representation of the structure of the experience. And the structural representations comes from the grid cells. When you think about the firing patterns, so the grid cells provide a metric representation of the environment. So the cells fire with a very regular pattern. And this regular pattern is maintained across different environments, different contexts. So this is an ideal coding regime to provide a generalized structured representation. Then, of course, you have the fast binding system, the hippocampal system with the place cells, which provide a representation of unique experiences. And the hippocampal system sort of is responsible to bind specific sensory experiences, which again come from lateral entorhinal regions into the system to these structural representations in the entorhinal system. And so bringing together sort of the general structure of environments with the sensory specifics via the hippocampal fast binding mechanism allows you to also flexibly recombining these representations. So abstracting structural knowledge allows generalization is one of the key assets which is computationally captured by the model and summarizing what we know about the different cell types. And factorization and conjunctive coding is also a very important property in the system. So one aspect what we always thought about is that these cognitive maps or cognitive spaces provide a domain general framework for understanding higher level cognition. And that's what I want to summarize in my talk with giving you some showcase examples of our work from different cognitive domains where we think that sort of the same representational principles underlie performance in different cognitive tasks. So let's first start with space and memory. So we often use virtual reality techniques in our experiments. Of course, we can't put magnets and scanners on participants' heads and let them navigate around, so we bring space into the scanner. We use virtual reality techniques, let people basically play computer games in the scanner and understand behavior and neural processes within that framework. So let me first talk about a study where we were interested in the context dependency of these mnemonic representations in the hippocampal system. So this is work done by Josh Julian, a previous postdoc in the lab who is now a postdoc at Princeton University working in rodent models in the Tank lab and in the Brody lab. So what Josh developed is a spatial navigation task, and you see sort of the schematic here, where participants have to learn different objects in a spatial environment. So you are exposed to different objects, and in the memory phase of the task, you're usually cued by an object. You have to go to the location where you picked up that object, press a button, and then you get feedback. So it's basically a virtual Easter egg hunt. And usually what we have is restricted virtual environments. It's an arena bounded by a wall, and we also have prominent cues, extra-maze landmarks, which provide orientation. And since we were interested in context-dependent representation, Josh used two different environments, a square environment and a circle environment. In both environments, we presented the very same four objects, but they were presented at different locations. So you see object one was presented over here in the square environment and over here in the circle environment. In addition to the difference in the shape of the arena, square versus circle, the two environments also differ in terms of the extra-maze cues, the landmarks around the arena. So you see them indicated here by the red and the blue dots around the arena. So it's different types of configurations of trees around the arena. In a critical third phase, test phase, we were exposing participants to an ambiguous environment. So we named it Squircle, so it's a mixture of a square and a circle. And in that environment, we also used all the extra-maze cues from the two original environments, the square and the circle. So you see those around here. And the critical difference to the square and the circle session is that in the square and the circle session, participants received feedback. So when they brought back the object to the remembered location, they received feedback of how far away they were from the correct location. We did not provide feedback in this critical squircle phase. It's a test phase. And there's also no correct answer. If you're cued by an object, you could either replace it, so the object one, you can either replace it here at the square location, or you can replace it here in the circle location. When we then look at the performance in the squircle environment, what you see is here at these, so these are response density plots where we present the responses of all participants for these four different objects in the squircle environment. You see that two response locations emerge. It's a bit like place fields. And these two locations are centered around the locations of the different objects in the two original environments. When you take object one, for instance, there is this square location which is over here and the circle location which is over here, and so forth for the other objects. So there's a clear distinction. People either replace a specific object in the circle at the original square location or the original circle location. And we can also look at the performance. So that's usually what we usually measure is the replace distance. So we measure the distance between the correct object location and the specific response location, average that across trials. And what you see here is that performance in the square and the circle is very similar. And also in the circle, here we, of course, there is no correct location, so we take the location, we take the location of the object which is, or we take the location of the environment which is closest to the response of the participants. So they either replace it next to the circle predicted or the square predicted location. So this is behavior. Let's look at the fMRI signal. So what I didn't mention, but that's the case for all studies apart from the behavioral only ones. We use fMRI, usually whole brain scans, acquire fMRI data throughout the cognitive tasks. So to understand this context-specific representation, what we did here is we trained a classifier, a correlation-based nearest neighbor multivariate pattern classifier in that case, to distinguish whether participants are in the circle or the square environment. So we're only talking about the original environments. And we can successfully decode whether people are in the square or the circle based on the pattern of hippocampal voxels. So you see that here, which is an indication of, you could call it a sort of a neural correlate of hippocampal remapping. I mentioned that before in my introduction about the place cells. When you bring the animal into a different room, the cell population remaps. So different cells fire at different locations. So there's basically an orthogonalization of the spatial representation in terms of the hippocampal place cell code. So we have a correlate of that here. Of course, you might not be surprised about that because also there are visual differences between the environments. I mean, despite the fact that we usually use the sparse environment, the X-ray cues differ, the shape of the arena differs, and so forth. What we plot here is just to illustrate the effect is what we call a contextual similarity score, where we plot for on average for each trial how similar the neural voxel pattern is to, if you like, a circle and a square template. And what you see is when they first they rotate in the environment, there the signal doesn't differ, but when they start to replace the object, recall the object location from memory, you see a sort of clear separation of the circle and the square and circle representation. So no big surprise, but we have context-specific representation in the hippocampus. Yeah? Interesting, it gets interesting now because we use that very same classifier where we distinguish circle from square representation and applied that classifier to the data in the third ambiguous environment, the circle. Of course, as I said before, there is no correct answer in that, or it's an ambiguous environment, there is no clear label. The labels for the classifier come from behavior. So whenever there is a trial where people replace the object close to the original circle location, that trial gets a circle label. If they replace it close to the square location, it gets a square label. So we can, based on hippocampal activity, classify in the ambiguous circle environment, and we are only talking about the very same environment, there are no visual differences, it's the same environment. We can basically predict whether people replace it or treat it as a circle or a square based on the pattern in the hippocampus. In many, many control analyses, and I can only summarize a few here, it is one of these papers with 20 supplemental figures. I want to highlight a few of the, I think, very important control analysis. So you might wonder whether this contextual similarity effect, where does that effect come from? So you could imagine that in the squircle environment, when you hang around the corners, because of the visual effects, you re-estimate the square representation, and likewise in other areas, the circle representation. But that's not the case. So there is no difference in this contextual similarity score depending on where in the squircle participants are. You see that here in these plots where we present the spatial distribution basically of the classifier output. So it doesn't matter where you are, it depends on your behavior, on your final behavior, or your final behavior predicts whether you reactivate the circle or the square representation, and not where you are physically at that specific moment in time in the environment, excluding sort of effects of sensory specifics here. We also do the very same, not for location, but for orientation. So you might wonder, okay, when I view the corner, I'm more likely to reactivate the square representation, but that's not the case. So there is no difference between this contextual similarity effect depending on the boundary view you have. And the same holds, you could also imagine, based on these extra maze cues, the environment-specific trees around the arena. When you look at a tree which was originally presented in the square, you're not more likely to reactivate the square representation. So what is the entorhinal cortex doing in this task? So we also looked in the squircle environment and in the original circle and square environment at entorhinal representation. So what we have developed many, many years ago together with Neil Burgess at UCL and Casper Berry is an approach where we look at correlates of grid cell-like activity in fMRI. Of course, as you all know, we can't image single cells with fMRI, unfortunately, but we can get a handle on properties which are very similar across a population of cells. In the case of the grid cells, it's certainly not location. We would never ever be able to decode location, I think, from entorhinal activity with a link to the grid cells, because the cells fire all over the place. But there is a systematic property across cells which is constant, that is the overall orientation of this hexagonal firing pattern. So in big clusters, in modules in the entorhinal cortex, despite the fact that the firing fields are shifted relative to each other, so you would have no chance to decode location, the overall orientation of this hexagonal pattern is similar across cells. So we take advantage of that property and look at a directional correlate of fMRI activity. And if it would be sensitive to a grid cell-like representation, it should have a characteristic modulation with 6 peak, a hexadirectional signal, we call that. So we are looking for these hexadirectional signals, so analyzing the running direction of participants in a direction-specific way, the fMRI signal, looking for a specific hexadirectional signal, a 6 peak modulation of activity as a function of running direction. So that's what Josh does initially for the circle and the square environment, and he sees a significant hexadirectional signal in entorhinal cortex in both environments. Interestingly, when you look, when you take the predicted hexadirectional signal from the square and plug it into the circle analysis, it doesn't work. So that means that the overall orientation of this hexadirectional signal, so if you like, the phase of that signal is different in the circle and the square, which makes sense, which you would also expect from grid cell recordings in rodents, because probably the grid cell system is driven by, or is anchored via the head direction cells to prominent extramaze cues. They differ in both environments, so the phase of this hexadirectional signal is different in both environments. Anyway, we have a grid cell-like representation in both environments. And then, following a similar logic of the hippocampal analysis, what he does then is, because we have this entorhinal context-specific representation in the circle and the square, he takes the phase of this hexadirectional signal and looks at every single trial in the squircle, again, depending on the behavior, whether people treat it as a circle or a square, to predict whether there is a context-consistent representation in entorhinal cortex. So if you like, the grid cell-like representation from the original square or circle environment is reactivated, versus a model which is inconsistent with behavior. And the model which is consistent with behavior predicts performance. and predicts fMRI signal in the squircle environment. So to wrap up, so we have some initial evidence for hippocampal remapping and, if you like, entorhinal realignment, which predicts context-dependent spatial memory in that study. Let's move on. I want to give you a second example on spatial navigation where we looked at, or we wanted to understand sort of the underlying representation by deforming the cognitive map, if you like. So this is work done by Jakob Bellmuth, the previous postdoc in the lab. And this idea, or that project started with some findings in systems neuroscience by Julia Kupich and John O'Keefe's lab, where they recorded grid cells in a highly polarized environment, a trapezoid. And what they see here is that the grid code seems to break down in the trapezoid. So when you look at the similarity of the firing patterns between, let's say, the two halves of a square, there's a high similarity between the two halves of this trapezoid, the correlation breaks down. When you look at gridness, that's a score, how you quantify the hexagonality of the firing pattern, that is also lower in the trapezoid. So something happens in this trapezoid to break down the regular structure of the grid cell code. So what we did is we used virtual reality in behavior. In that case, we were really interested in physical navigation. So we used this so-called virtualizer. If you like, it's a bit of a cheap variant of a tracking lab where you let people navigate around, so you can use it in small rooms. So they basically move on that platform, which is nice because it's omnidirectional. It's not like a treadmill, and you can turn around. So that's the setup. Task is very similar to that what I described in Josh's study. People have to memorize different objects, bring them back to the location, get feedback for it. We used 2 sessions. In one, we exposed participants to a square environment and another to a trapezoid environment. And we carefully controlled the location of the objects. So there were 6 objects in total in both environments, and they were arranged in, unknown to participants, in a very specific manner because we wanted to keep the object-to-object distances in the 2 halves of each environment and in the 2 environments constant. It will become clear later why we did that. So first we looked at spatial memory. We normalized, or we quantified memory by also taking into account the possible difference between the error distributions in these differently shaped environments. And memory is better in the square. So participants are significantly better in the spatial memory in the square compared to the trapezoid, taking into account or controlling for the differences in error distributions in the 2 environments. And they are also worse in the narrower part of the trapezoid compared to the broader part of the trapezoid. So you see small but highly significant behavioral differences between the 2 environments and then within the trapezoid environment. Then we wanted to link this purely behavioral study more with what we know about grid cells. So what we did is we modeled a population of grid cells using the successor representation, and this was worked in collaboration with Caswell Berry and his postdoc Will Decotti, who did all the modeling for us. So they model a population of grid cells and let basically a virtual rat navigate a square and a trapezoid environment and use the predicted grid cell population activity, and they replicate with their model the findings from the Kupich study. So when you look at the similarity between the 2 halves of both environments, there's a higher similarity of the grid cell of the model grid cell activity in the square compared to the trapezoid. Then what we didn't expect, so this is a discovery we made by just applying this model, is we also looked at the spatial frequency of the grid cell firing pattern, the model grid cell firing pattern. And we see a systematic difference in the spatial frequency. And with spatial frequency, I mean the regularity, the spatial frequency of the grid cell, grid fields basically. So in the square compared to the trapezoid, you have a higher frequency of the grid pattern. So in the square, the cells are more packed compared to the trapezoid. And we also see that difference between the broad and the narrow part of the trapezoid. And sort of the translation from that finding to human behavior, the prediction would be that based on the systematic difference in the spatial frequency of the modeled grid cell firing pattern, we would predict larger estimates in the square compared to the trapezoid when people have to estimate distances, and the larger estimates in the narrow compared to the broad part of the trapezoid. So how did we measure that? We measured it in two ways. So in one session, we presented participants always the images of two objects, and they had to navigate physically the distance between the two objects. So that is a sort of navigation distance estimate. And we also had a desktop-based version of that, where people see the two objects, and on a scale they have to indicate the distance between the two objects. And now, of course, the highly controlled distances between objects becomes relevant now in the analysis. When we do that, and I only show here the data for the walking test, but the desktop test looks very similar, we basically predict the model, we confirm the model predictions. So we see an increased distance estimate for the square compared to the trapezoid, and for the narrow compared to the broad part of the trapezoid. So the predictions coming from the grid cell model in terms of the spatial frequency, so how packed the grid cell firing pattern is, directly translates here into human behavior when they explore a square in the trapezoid environment. So to sum up this finding here, deforming the metric of a cognitive map distorts memory. Let's change topic and talk a bit about time. As you all know, space and time are highly important for episodic memory. We only have an episodic memory of a specific event if we remember the location, so this room, for instance, and the specific time. It's an important difference whether it's today or the meeting in 2019. So this is work, ongoing work actually by Ignacio Polti, who is a PhD student in the lab, soon going to be a postdoc with the Moses, switching techniques and becoming a systems neuroscientist. So we used, in that task, it's not about episodic time. I'll briefly mention another study where we do that. We use a time to contact paradigm. So participants see a moving dot, boring experiment throughout the session. They see a moving dot, and that disappears at some stage, and they have to indicate when the dot hits the wall. And unknown to participants, we nicely sample the entire directional range with a highly controlled trajectory throughout the task. And unknown to participants, but they become aware of that, we manipulate the speed. So we have 4 different speed levels. And when we look at performance, we replicate sort of classical time-to-contact TTC effects in the literature. So when you plot the correct, so the target TTC against the estimated TTC, you see that people overestimate the short TTC, so they think they take actually longer, and the long TTCs they underestimate, so they think it takes less long, and you see this sort of this effect, this trend towards the mean. And I have here some plots from example participants where you see how that develops over time. So people over time adjust basically their performance reflecting this grand mean of the TTC distribution. Yeah? And do we have some weird? feedback, yeah, doesn't matter. So we can also quantify behavior with a measure of, or with precision accuracy measures. And what you see is that performance is actually best for that speed level, the third one, which mimics the average of the entire distribution. That becomes relevant in a second. So in an initial study, which we already published, where we're interested in sort of the basic learning effects, feedback-based effects, finding effects in the hippocampus. In that specific study, I want to focus on entorhinal cortex. So what we did here is similar to that what Josh did in virtual reality, where we let people navigate and analyze the fMRI signal as a function of the virtual running direction in the environment. We here analyze the signal in a very similar fashion, but it's not the virtual running direction which we're analyzing, but it's the gaze direction. So because we have this highly controlled gaze trajectory with the moving dot, we can analyze these data in a very similar fashion. So look at this hexadirectional signal as a function of, in that case, the gaze direction of participants. And when Ignacio does that analysis, what he sees is that, interestingly, the hexadirectional signal in entorhinal cortex is highest for that speed level which is closest to the average speed level in the experiment. And we can also look at other measures. We can look at the spatial stability of the signal. So that means how stable so we look at the entire entorhinal cortex, and we look how consistent the signal is across voxels. So the spatial stability is very similar between the different speed levels. Interestingly, when we look at temporal stability, and that is then how consistent the entorhinal signal, so in terms of the phase of this hexadirectional signal, is across, for instance, 2 sessions of the experiment. And there, the temporal stability is interestingly highest for, again, that speed level, which mimics the mean of the distribution. Ignacio then also links that signal to behavior. So when we take that measure, this precision accuracy trade-off measure, that correlates with this grid-like signal here in that specific speed level. So the stronger that signal is, the higher performance is. And Ignacio also uses some modeling, some Bayesian observer modeling, where he then takes the prior in the model as one of the 4 speed levels, and the model fits the data best when you take the 3rd speed level, so the mean across the distribution, as your prior in the model. And that explains behavior, and also the model predicted effect on behavior also correlates with this grid-like signal in entorhinal cortex. So in sum, this study provides some initial evidence for grid-like signals in entorhinal cortex, which are modulated by the regularity of the temporal context. So somehow the entorhinal cortex seems to be sensitive to structural information of the temporal context. I want to give you another example, also in the time domain, very briefly, on episodic time. So when you, for instance, want to remember on the next day when your mother called you, you might remember that it must have been somewhere between the start of the football match you were watching on television and the time you went to bed. So you, based on these on your episodic memory about other events on the time, you can sort of reconstruct the time when your mom probably called. And Jakob Bellmuth, the previous postdoc in the lab, also investigated this effect in a study. It's also an fMRI study. Again, I don't want to go into detail of the study. I want to share one behavioral effect with you, which mimics the one from Ignacio's study. But I need to briefly introduce the task to you. So participants were exposed to a whole bunch of scenes from a virtual family. So we presented different snapshots of different scenes, So for instance, somebody is doing the washing up, there is a guy reading, and so forth. So you have different images describing the life of a virtual family over different days. So we had in total 4 different virtual days. They also differed in terms of the length. They will have shorter and longer days. And we were interested in how the hippocampus especially encodes temporal information in that task. Behavior, the critical behavioral measure is measured in a timeline task, where after the experiment, we expose participants to a whole bunch of snapshots, and they have to drag these objects to the correct time. And so this is a schematic of the 4 virtual days in terms of the different length of the task. So what we now did is, because every day had 5 virtual events, we looked for a specific event. Let's say it's the 4th event of a specific day. We were interested whether the temporal memory of that 4th event of day 3, for instance, is influenced by your memory of the 4th event of the other days. And so we calculated the influence of the other 4th events on the other virtual days on that memory of that specific event. And what we find, just to cut a longer story short, is we find a correlation between the constructed virtual time, which seem to be overestimated when the events occupying the same sequence position in the other sequences took place late relative to the event in question, and vice versa for an underestimation for those where it took place very early in the sequence. So your memory of this sequential position in the other sequences seems to bias your performance, and we see that in all participants, and the effect also correlates with the hippocampus signal, so how well you represent temporal information in the task. Just wanted to briefly highlight that because we see this sort of similar generalization effect in this very sort of psychophysics-like time-to-contact paradigm and in that more episodic memory task I just described. So last study I want to briefly present is a study done by Mona Garber in collaboration with Nico Schuck and Eric Schulz from 2 other Max Planck institutes. So in that study we were interested how these cognitive maps might support decision making. So a complicated experiment where participants were exposed to a whole bunch of monsters in a virtual reality environment. And in the first session, was a spatial memory task where they had to learn the locations of the objects. And then in a choice task in the scanner, we trained people to associate rewards to different objects. Unknown to participants, in 2 different contexts, the rewards were following a spatial distribution. So imagine you have learned these 16 objects in a spatial environment. And then you are exposed to these objects later on outside the spatial context, but the spatial proximity determines the reward value. So all these objects which are in red here are highly rewarded, but they appear all in the same area of the arena. And we have 2 different contexts. In context 1, it's those objects which are rewarded. In context 2, it's those objects which are rewarded. Critically, we also have 4 so-called inference objects, 1, 2, 3, and 4. Those are objects which are never rewarded, but we would predict that object 1 would be associated with a high reward because it happens, it was originally learned in spatial proximity to other highly rewarded objects compared to the object 2. And the same holds for object 3 versus object 4. So let's first talk about behavior. This is actually indicating, this plot E here is indicating this inference performance. I might overrun 2 or 3 minutes. So when you plot these different inference objects, 1, 2, 3, and 4, and in a final task of the experiment, they had to estimate the value of the objects. Despite the fact that object 1 and 2 were never rewarded, they associate object 1 with a high reward and object 2 with a low reward, and the same holds true for the other context. This effect, so we can also quantify this inference performance or inference error, if you like, if you turn it around, and that correlates with your spatial knowledge. So participants who have the best spatial knowledge of the environments are also those who have the highest inference performance in that task. So it becomes interesting now because in that experiment, unlike many of our other studies, we let participants completely freely choose these objects. So they have a learning session, they are exposed to these objects, they can choose whatever order they want to sample these objects. And you see here 3 example trajectories from 3 participants, and you see the stark differences of how people sample these objects. We can also have a graph representation of that. You see the sampling strategy is completely different between participants. And then we joined forces with Eric Schulz and his PhD student Tancred Sanum, and they applied a Gaussian process model on those data. And what the model can basically predict rewards for a novel stimulus based on the reward associated with all other stimuli, and weighted by their similarity to the novel stimulus. And this similarity can be quantified based on 2 sources of information. You can take the ground truth, which is the spatial location, but you can also take temporal information into account. And temporal information we get from the very first phase of the experiment, where we analyze the temporal sampling of objects by participants. And when we look at these different kernels, so the spatial and the temporal, or we can call it predictive one, the combination of both predicts best behavior. So information about the spatial distribution of objects, but also how you individually sample them in time in the initial phase. The model can also nicely predict the performance of these on these inference objects, and both the actual inference performance correlates highly with the model predicted inference performance. We can also show that the spatial predictive kernels predict best the performance of the value weighting compared to a spatial only or temporal only model. And these 2 effects actually correlate negatively across participants. So participants who were relying more on a spatial map rely less on this predictive or temporal map, and vice versa. And spatial, but not the temporal kernel of the model, predicts the actual inference error. So the spatial information correlates better with the inference performance. Last slide, I promise. It's only on to give you some overview of the neuroimaging findings. We then can also look at spatial and temporal coding in the brain. And for the spatial case, we take fMRI adaptation as a proxy of how people encode locations in the environment. So spatial adaptation scales with the distance between objects in the remembered objects in the environment. And this spatial fMRI effect correlates with the spatial effect on the choice behavior as modeled by the Gaussian process model, and also the spatial fMRI effect correlates with this inference performance. Over time, participants, and here you see the data from all participants, the spatial weight takes over. I mean, at some stage, the ground truth sort of comes true, and the degree of at which that takes place, so the slope of that function, the more the spatial information takes over behavior, the higher the slope is. The slope of this effect also correlates with the inference performance, and also the slope correlates with the spatial fMRI effect measured in the fMRI session. So to wrap up, the key finding of that study is that hippocampal cognitive maps seem to adaptively guide reward generalization. It looks like that we have several map-like representations in parallel stored in the brain. One is the spatial one, the other one is the predictive one in that case. And let me jump to my Outlook slide. So just want to wrap up the findings I described here. You saw some evidence for context-dependent memory representation showing remapping in the hippocampus system, alignment in the entorhinal cortex, which predicts context-dependent behavior. I showed you an example on deformations of spatial representation, the conceptual ones I skipped in that talk. I showed you how the system encodes the regularity structural information of the temporal context, and the last study demonstrates some evidence for the relationship between cognitive map representation and reward generalization. I want to just provide some overview. This is actually for the final discussion now. Some important points we can discuss. Let me, I summarize it in a second actually. I just want to thank my lab. This is just an overview of the different people and my funding agencies, or Max Planck Society in particular. And with that, I actually jump back because I think that might be a good starting point for general discussion. Thanks very much.
Thanks, that was great. I was intrigued by your findings with the trapezoid, comparing that to the square. I was wondering why the frequency of the grid cells would differ in the narrow end and the broad end of the trapezoid. And is that related, could there be a relationship with distance? So if you see a square in perspective, it's essentially a trapezoid. And so is that why you looked at trapezoids in the first place? And is that the hypothesis, or is that?
The funny thing is we did not predict that, so it was an observation based on the model. We also then, because Casper Berry was also involved in that Cooperage study, I think he also looked at the data, and I think it was too noisy to see that in the actual cell firing. A reason, and we of course model a large population of cells, that's why we might be sensitive to that effect. A reason could be, we don't know actually why that is the case, a reason could be uncertainty. So what we know is that the grid cells adapt the scaling based on the uncertainty. So it could be the case maybe that the uncertainty is higher in the trapezoid, but we actually don't know. I mean, it's just a prediction coming from the model, which would but then directly relate into that sort of behavior prediction. Yeah, but it's a
but do you think there is a relationship with distance? So if you are in an environment and you have to judge the distance between locations far away or nearby, which becomes essentially like a trapezoid.
I mean, this is a reasonable interpretation, because that's what we actually then effectively measure in our behavior. But whether that's the case for the rat navigating around, we don't know. Yeah. I mean, this was one of these situations where you use a model and that predicts something which you haven't really expected. But is somehow interestingly reflected in the behavior.
Thanks, Christian. That was really interesting in terms of, I understand the general approach of building from the grid cells to cognitive maps that we could then generalize across lots of tasks. But when it comes to the hippocampus, as you know, there's more and more evidence suggesting we should not think of this as a homogeneous structure, and that there are clear differences that seem to be important for both functionally and in terms of connectivity. For example, and whether, well, number one, have you looked at those kinds of issues? Does it matter? It looked like some of your data was more biased towards anterior hippocampus, for example. But the general question is, have you tried to address that issue, given the kinds of data we now see about compartments within?
Yeah, very good question. In the spatial context study, the analysis was done on the entire hippocampus. We did not separate between anterior and posterior. In the episodic time study, where I only highlighted the behavior, because that I thought was an interesting sort of generalization effect we see there, linking to the other time study. In the imaging part of that study, we see the sort of, so the key effect what we see in the hippocampus is that the hippocampus seems to be sensitive to the time within day. So when we look at representation similarity and fMRI adaptation, that scales with the temporal distance of events between between events of the same day. And that effect is strongest in the anterior hippocampus. Yeah, so there we separate between anterior and posterior hippocampus. And in all our previous temporal memory studies, we usually see the strongest effect in the anterior hippocampus. With long axis distributions and lateralization effects, probably sort of the pure spatial function is residing more in the posterior hippocampus, with exceptions. We had one study where we explicitly looked at long axis distribution of effects. That was an episodic memory study almost 10 years ago now, where we tested the idea that different events which belong together, so in these memory integration paradigms, for instance, the idea was the bigger these mnemonic networks are, sort of the more events you have to link together, the broader this mnemonic network gets, the stronger the anterior and pericampal representation should be. And we see basically a scaling of the size of these mnemonic networks along the long axis, with the smaller mnemonic networks residing in posterior hippocampus and the bigger networks in anterior hippocampus.
Thank you. So I had a question about the generalization to conceptual representations and more cognitive representations. And in the context of space, the axes or dimensionality is given and fixed by the two-dimensional layout of geography. And in the context of time, it's also given in a sense. But the example of, the motor vehicles, there's, obviously an indefinite number of dimensions along which you could sort those items, and you represented two. But where do the dimensions come from to the hippocampus? So if it is applying a sort of grid-like representation to concepts, how does it know which dimensions upon which to apply that grid structure? And is the magic trick already done by the time it gets those dimensions? And is it really sort of structuring those dimensions if they're already given to it?
It's a super interesting question. I can show you one slide, actually. What we have done, and others in the lab, also including actually a work by Manuela Piazza from GMEC, is presenting low-dimensional feature spaces to participants. So just give you an example. So this is the example from the Constantinescu study from Tim Barron's lab, where they used a bird space, where they manipulated different features of the bird. We used in studies done by Stephanie Teves in our lab, for instance, circle size and opacity, and let participants, in the original Barron studies was pure feature space. We use a categorization task, so people have to learn concepts. So they associate category A with all those feature combinations over here and category B over here. What we and others see that the hippocampus and the entorhinal cortex seem to be sensitive to the two-dimensional nature of the task. And you're absolutely right, that's what the experimenter gives to participants. In one study by Stephanie, we looked in your questions in a little more in detail. So we used the three-dimensional feature space, but at any given time, only two dimensions were task relevant for that categorization performance. And what we see is that the hippocampus signal is adjusted to only those two dimensions which were task relevant. So if we sort of what I think is going on is, of course, as we all know, the world is multidimensional and conceptual information is multidimensional. It could well be the case that the hippocampal system, tuned to navigate in 2D, sort of carves out the task-relevant dimensions and provides a low-dimensional representation of that higher dimensional space. So that is, I mean, we don't really have experimental proof for that, but with that one study, it points a bit into that direction, yeah.
Thank you for a very inspiring talk. I was, of course, based on my own conceptualizations of anchors and stuff, I was wondering in your spatial context, and I think it was one of your outlook questions that you put in there. So how flexible are those spatial representations? Because I was always wondering with the firing rates of grid cells, right? They're really based on XY coordinates almost of that space that you encompass. And I was wondering if this kind of whole grid could basically morph based on what I would call anchors, or you called landmarks of things that are surrounding it, where you know, when you come into any kind of room, it might not always be the far right that is important for food, but it might be dependent on where the food dispenser is in that room. So how basically, how flexible is your spatial representation in morphing along and changing its direction and maybe, you know, form and shape depending on where other important objects are that you might have prioritized due to reward, for example, as you've shown, like the distance or the proximity to those reward locations might be important. What is your take on that?
Very good question. Talking about reward, what we know is that the grid map is deformed around reward locations. So that is evidence from rodents. Simone Vigano, who's sitting in the room, has a study in humans where he shows also evidence for that. So that about reward. You're asking exactly the right question. So what the entorhinal cortex provides is structural information. When you think about sort of going to your favorite supermarket in Frankfurt and then going to a supermarket in Rovereto, your grid cell system should encode the structural regularities across these different supermarkets. So that is a schematic structural representation. And then the hippocampus, in concert with lateral entorhinal cortex, which gets all the sensory information in, you encode the sensory specifics. I mean, your very specific features in your Frankfurt supermarket and then the Rovereto one. And the key thing is how are these sensory specifics combined with that structural information? And that is not trivial when you think about it. And what at least the Thomann-Eichenbaum model by Tim Behrens proposes is that, and that reminded me a bit of your ideas about anchors, is that maybe the object vector cells, so these are cells which encode your allocentric distance and direction to objects. Maybe it's that type of coding, sort of coding something which is in between sensory specific information and pure structural information, which then anchors this grid map across to the very specific environment. So, but this is exactly the thing, what is very important to understand. How can this rigid structural code be aligned, adjusted to the sensory specific, to the episodic information you're encountering, and sort of how is the alignment done correctly, basically? Yeah.
Exactly, and how does this abstract representation look like, right? That you can then kind of apply to specific environments that you might be dealing with. Mm-hmm.
I mean, there's cognitive psychology probably has the answer as well. I mean, there's famous structural alignment theory and so forth. But how that is done specifically, and when you really think about it, it's not that easy to get your head around it. And maybe that information from the object vector cells is, at least in terms of cells, probably the type of coding which provides that alignment.
Yeah, going back to the question, one of the previous questions, how would you, I'm here, how would you relate your work to work of Fulford, Quiroga about concept cells, that specificity of the particular instances is more specific from the first visual field along the ventral stream toward the hippocampus? That would be one part of the question. And the other is like if we go even further toward the anterior portion of the temporal lobe, temporal pole, there are also examples of very specific individuals even coded in that. How this axis would be related to the hexagonal grid cells? Mm.
That's a very good question, and the general question, I guess, is how is that what I was talking about related to long-term semantic knowledge? Yeah? And what we usually look at is the acquisition process, and that's what we're interested in. How is new structural information, new knowledge acquired? And I think in that acquisition period, the hippocampus system is crucial. The long-term storage is, of course, happening outside the hippocampus. So understanding that interplay is, I think, also very important. Yeah? But I think the system is in particular important in the acquisition stage, to acquire new knowledge, to make these new links, to acquire new structural information. I didn't answer your question about the concept cells, but I'm not quite sure what that finding means. There's a very specific information in single cells. I think I try to think more in terms of the population representation.
Yeah, like this terminology is very unfortunate. Like it goes from Konorski, for example, but like we can think about these cells as populations. Obviously, like the recordings from single cells in the epileptic patients are very vivid, so like we see the activation or no. But the fact that we see activation of certain cells do not exclude that there is a population of cells that are becoming activated. And for example, rodent literature from Tonegawa laboratory indicates that there are engram populations, that the whole population encode a given memory. And think about the encoding information. Like in these experiments on concept cells, they present, for example, new pictures and see how these pictures are recognized along the ventral stream. So these are also new encoded information, so to speak.
So I'm also interested in how we can extend these ideas to the conceptual domain. So it was really interesting how you showed that the grid cell maps deformed in the trapezoid shape relative to the square shape. In the spatial domain, we know exactly what shapes those are, right? Because a foot in one direction is the same as a foot in the other direction. So we know one is square and one's trapezoid. But for conceptual dimensions, we have no idea what shapes those are. And they're very likely not square, right? Because we have no idea how warped these different dimensions are in the brain. So if it's true that non-square shapes lead to deformed spatial representations, then to what extent should we expect the system to be able to support the representation of conceptual knowledge?
Yeah, when you think about it in terms of updating conceptual representation, integrating new exemplars into your conceptual representation of a specific theme, you could think in terms of also squeezing and stretching your conceptual spaces to some extent. So the spatial analogy, I think, would probably still hold.
So, so context-specific warping of the space to create a square in every situation?
Yeah, I mean, yeah, of course, this is due to the nature of the experimental task we're using. I mean, in terms of a low-dimensional projection of conceptual representation, you can think along the lines of deformations of these conceptual low-dimensional representations to integrate new information, to update information. Yeah.
Just to follow up, so I was wondering, have you looked at frontal cortex in that context, in the Squircle experiment? Could it be that it would be predictive of the schema that would be applied to each of the environments?
So you're talking about the
Squircle.
Yeah, we are often, unfortunately, hippocampus centered and only look at that system. We have not really examined the prefrontal cortex in great detail there.
Thank you.
Great, thank you again. It is time for lunch. Please be back here promptly a couple minutes before 2 so that we can start the next talk right at 2. All right.